السلام عليكم. شكرا نشكركم على الحضور. طبعا في البدايه يعني وقت كان يمكن غير مناسب، ما كنا ماخذيها بنظر الاعتبار شغله الزياره، بس الحمد لله ان شاء الله البركه بيكم. وايضا المحاضره راح يتم تصويرها فعلى مود الفائده العامه للاخرين، فشكرا جزيلا دكتور ضياء. طبعا راح اتكلم اليوم بشكل عام على الانتيبودي بيست ثيرابيوتكس. راح البرزنتيشن حتكون تقريبا 60 داتا 50 باك جراوند. راح اتوسع بالباك جراوند لان شويه يعني الفيلد اتس نيو فيلد فيحتاج باك جراوند. ف let's start with a science prank يعني or humor. ف what do you call an acid with an attitude? Give me your lunch. Amine acid. So it means, actually I showed this slide to my wife. شوفت لزوجتي عليها فاخذت رايها بي، قالت ليش ما تشوف ولاية بطيخه مثلا لك. اوكي فعادة يعني برزنتيشن نحب نبديها في شيء لطيف. طبعا الامينو اسيدز هي تعرفون مثل تعرفون هي البيلدينج بلوكس اوف ذا بروتين ثيرابيوتكس or biotech products in general. And there are really some mean amino acids like جدا يكون problematic و difficult بالديزاين of your protein drug. قسم منها يعني تسوي لنا مشاكل هوايه، قسم منها more susceptible for like protein oxidation. Asparagine, for example, is highly problematic and is susceptible to deamidation, which is a big problem in the stability of protein therapeutics. So, why antibody-based therapeutics are important? So if you take a look at the top 10 selling drugs, you'd find 6 of them are antibody-based therapeutics. For example, if you take a look at the best selling drug, which is Humira by AbbVie, it's 14 billion. This is a comma. It's actually not a period. So it's 14 billion, and that was in 2015. And the market is growing fast in a fast pace. So 85 billions in 2015, and it's expected to reach around 140 billions by 2024. If you take a look on the second biologic, they call it, by the way, so antibody-based therapeutics also it has many names, many synonyms. Sometimes it's a part of biologics in general. You have biologics versus small molecule drugs or traditional drugs, and sometimes they call biopharmaceutical products and protein drugs. So it's different since it's new relatively new field. So you have many terminologies. Enabral, which is also an antibody-based therapeutics, it's actually just the Fc part of the monoclonal antibody, and it's fused with TNF, tissue necrosis factor receptor, so that it acts as a trap for TNF, and it's used to treat rheumatoid arthritis. It was approved by Amgen and Pfizer, and again you see it's rank 3, and then Remicade by Johnson & Johnson, and Rituxan by Roche. It's Genentech actually who developed Rituxan. And then Genentech, which is the birthplace of biotechnology, as I'll show you later, also was acquired recently by Roche. And then you have Avastin, of course, you know Avastin for treatment of AMD, again by Genentech, and trastuzumab, Herceptin, again by Roche. And if you take a look at the small molecule drugs, it's like Harvoni, which is the second best-selling drug, or second blockbuster, at least in the United States. So this small molecule drug is an exception. It's an exception to other small molecule drugs. It was developed by Gilead, and it's used to cure actually Hep C, virus. So So the reason why it's best selling, it's because the price of this drug is so expensive. One tablet costs about $1,000, and I think it's like three months course of treatment, so it costs about $80,000. So if you produce, and India, of course, produce Harvoni, and they sell it for cheap price. So, and again, if we take Harvoni out of this list, then again, you would be left only with biologics or antibody-based therapeutics. Antibody-based therapeutics, of course, these are monoclonal antibodies that we are talking about. And then these are just some examples about the antibody-based therapeutics. So usually most of the monoclonal or the antibody-based therapeutics are just conventional monoclonal antibodies, like these guys, Humira and Remicade, and etc. Also, you have what's called Fc fusion. So people also fused the Fc region, and I'll tell you why in my next slides. Fuse Fc fusion with some other proteins. So they call it fusion proteins to make therapeutic with desirable pharmacokinetics properties, like Enbrel, which is for treatment of rheumatoid arthritis. Also, people use the Fab region, just the Fab region, to make some antibody-based therapeutics. And the most famous one is Lucentis, which is anti-VEGF, vascular endothelial growth factor receptors, and again, it's used in the treatment of AMD. And another big field that's evolving right now is the bispecific monoclonal antibodies. So for the bispecific, you have people also engineered some antibodies to have bispecificity. Instead of the monoclonal antibody, you have two identical Fab regions. And then bispecific, you have different Fab regions. One Fab is specific for, like, say, tumor cell, and the other Fab can bind specifically to a T cell, so that T cell can help in eliminating of this tumor cell or the antigen. And of course, antibody-drug conjugates is a big area right now. And people with antibody-drug conjugates, people try to conjugate some small molecule drugs or some toxins, they call it payloads, at certain regions on the monoclonal antibody, so that the monoclonal antibody would deliver this toxin into the tumor cell in a specific manner to reduce the off-target toxicity. And it's a very complicated technology, ADC, and there are only two ADCs that were approved by the FDA, and many others are in clinical trials. And even AAC, antibody-antibiotic conjugates, they take antibody and try to conjugate antibiotic instead of just toxin. So there are some bacteria that methicillin-resistant Staph aureus. Recently, they found that methicillin-resistant Staph aureus hides inside the cells. So the only way to deliver the antibiotic to the inside of the cell is through the antibody, which can be also pinocytosed by the cell, and then the antibiotic would get released inside these cells to kill the hidden infection, and so on. There are many other antibody-based functionalities, and people are using fractions, another fractions of antibody to make specific antibody-based therapeutics. Then, if we take a look at the market share by indication, so most of monoclonal antibodies are actually indicated for cancer, to treat cancer. And cancer is a big area. It's a wide open area, and it's very challenging to develop an anti-cancer drug that can cure cancer and prevent any relapses or whatever. Another big area in the monoclonal antibodies market is the autoimmune disease. Rheumatoid, as I showed you in the first slide, Humira. It was used in the treatment of at least rheumatoid arthritis and some other diseases. And then infectious diseases, it's taking another big chunk of this pie chart. And then, microbial diseases, antibiotic conjugates, as I showed you, and some other inflammatory diseases. Now, so before going too deep into the research data, I just wanted to show some historical aspects of the development of antibody-based therapeutics. So the first people who described the antibody structure were Gerald Edelman and Randy Porter. So those guys in 1961, they collaborated and they put a full description of the antibody structure. So this guy actually, he said, or he figured out that the monoclonal antibody is composed of 2 light chains and 2 heavy chains, and these are interconnected through disulfide bonds. While the Randy, he described, he found that the antibody is composed of 2 Fab regions, and these are identical regions, and 1 Fc region. And he used enzymes, certain enzymes, to conclude these results, and then they collaborated with each other, and then they published and they patented they published the first structure of the antibody, and also they got Nobel Prize in medicine physiology in 1971 for their description. Now, you might have asked, how do we make monoclonal antibodies? So the first technology to make monoclonal antibody was called the hybridoma technology, and people in the biochemistry department are pretty much familiar with this technology. So it was developed by Kohler and Milstein in 1971. So what they did, those guys, again they got also a Nobel Prize. So they took an antibody-producing plasma cell from a mouse, and then they fused it with a cancerous plasma cell, with a tumor cell, and they used chemical means to fuse these cells, and the results of this fusion is what's called an immortal cell. It's a hybridoma cell, and the hybridoma gives you the desired monoclonal antibody that was first produced by the antibody, the plasma cell or the B cell of the mouse. So people used this technology to produce some monoclonal antibodies, usually for diagnostics. And actually the first approved monoclonal antibody, which is the orthoclone, was also produced by this technology, hybridoma technology. And however, this orthoclone, which was used for the treatment of kidney transplant rejection, it had a mouse IgG sequence, which is the IgG2A. And mouse amino acid sequence, these are usually recognized as foreign bodies by the human immune system, and they have a lot of immunogenicity problems. Later on, the recombinant DNA technology kind of like replaced the hybridoma technology for the production of monoclonal antibodies. And recombinant DNA technology was actually born on a deli napkin. So it's like a mendil مال أكل. شلون أكو 2 scientists, Boyer from University of California, San Francisco, and he collaborated with Cohen, and they they had a meeting at a restaurant, and then immediately they shared ideas, and then they took a napkin, and then they first described their recombinant DNA technology on that napkin, and after that they patented it. And then Boyer, one of them, Boyer, he met, like a few years later, he met Swanson, a venture capitalist, and then they agreed to establish a company called Genentech And Genentech actually is the birthplace of pharmaceutical biotechnology. It's a great company, and it was the first company that produced recombinant DNA protein, which was insulin, with the collaboration of Eli Lilly. And then most of the products that I showed you in the first slides are from Genentech, which was later on acquired by Roche. And then, so that was this. And then the recombinant DNA technology. So you might have asked, then recombinant DNA technology was described in 1974, something like that. So why in 1986 people still using hybridoma technology? So the reason why the first description or the first protein or insulin was made in using E. coli. E. coli is, Escherichia coli. It's an eukaryote organism. It's not that well developed. It's okay. It can give you insulin. It's not that complicated. However, if you want to make an antibody, monoclonal antibody in E. coli, you can't do that because it's beyond its capability to form the proper disulfide bonds, and also beyond its capability to put the sugar part, or what's called N-glycan, on that, which is an essential part of the monoclonal antibody. And I'm going to talk a lot today about this part and what we did with the sugar part of the monoclonal antibody. Now, if you look at the aspirin, which is a small molecule drug, so the size of aspirin is actually about 180 Dalton. So if you compare it with a full-length antibody or monoclonal antibody, IgG, we usually use companies usually, almost all of the monoclonal antibodies, therapeutic monoclonal antibodies, are of IgG isotype. They don't use IgD or IgM or the other classes. Only IgG is used to produce therapeutic monoclonal antibody. So again, the size is 150, is extremely big compared to the aspirin, and sometimes people like to compare aspirin with the bicycle, and then insulin with the car, and then like the monoclonal antibody with a jet or airplane. So the problem with these biologics or biopharmaceutical products, the problem with these products is that they have in addition to their primary structure, which is just the amino acid sequence, they have a secondary structure, alpha helix or beta sheet, or some other secondary structures, and then these secondary structures fold into 3D structures, the tertiary structure, and you can also go to higher order structures, the quaternary structure, when protein domains associate with each other. And again, the problem with these big biologics is that they have, in addition to the covalent bonds, they have non-covalent bonds. And by the non-covalent bonds or interactions, you have many other important essential interactions, the hydrophobic interactions, and you have also some salt bridges and have hydrogen bonds, and these are very essential for the protein proper conformation or the native conformation. Because one of the biggest problems in making protein therapeutics is the aggregation. Aggregation proteins, are very dynamic because they have weak interactions. They are not solid molecules. So sometimes under certain conditions, like elevated temperatures, these proteins start to unfold, or usually the inside of the protein is hydrophobic and the outside is hydrophilic. So when they start to unfold, they expose hydrophobic patches, and then the neighboring protein molecule also expose the same hydrophobic patches, and then there is going to be some sort of hydrophobic interactions to form a nuclei, and then this nuclei then can grow up into small aggregates, larger aggregates, and they can go into the precipitate. So and the the critical thing about aggregation is that most of these aggregates are highly immunogenic, and they pose a lot of safety problems. So, and if you look at the development of new chemical entities or small molecule drugs compared to a biopharmaceutical or biologic, you see it's the type of the number of the documents that are required for the approval of these biologics are huge. It's a mess compared to a small dossier for the small molecule drugs. So this is one of the favorite slides. People always use it. And then, so if we compare the biopharmaceutics or biopharmaceutical products or biotech products to the small molecule drugs, so the manufacturing processes are a little more complicated than those. So usually with living cells, as I said, E. coli, like yeast, and also mammalian cells give you more complicated proteins. So these are difficult to control their growth, their secretion of the protein, and also physiological temperatures are you have to keep your proteins refrigerated or cooled so that it doesn't get unfolded and aggregated. Production time usually takes months. These cells takes a lot of time, especially mammalian cells. They take a lot of time to grow up, and then you start the induction phase to produce the protein or your therapeutic protein, and you have to monitor the process, stringently, and you have about more than 30 because production of these biologics takes about more than 30 operations compared to only 10 units operations in case of small molecule drugs. And again, you have a lot of end process control points, and then extensive environmental monitoring. Most of these biologics are given through the IV route, or sterility-wise, you have to be very careful. And the other important issue with these biologics is the viral contamination, because these cells usually, if your cells that gives you the protein are already contaminated with some viruses, or the other nutrient like the calf serum that you use to feed these cells is already contaminated with some viruses, and you don't do a good job in decontaminating these products, then imagine these products will go through a cool chain, and then viruses can still alive in these throughout all of this distribution chain, and then when it reaches the consumer, eventually the consumer will get infection. So and also the complexity of these products, the biotech products, also requires a lot of product characterization. For example, for the quality control analysis, you need about 30 tests to make sure that your product complies with the specifications. And again, as we said, these are usually administered by IV or sub-Q route. And then if you change the process only a little bit, then you get different glycoprotein or protein. And then another important point about this biologics is that expiration date usually has to be calculated in real time. Stability studies, not an elevated temperature stability studies. Arrhenius usually does not predict the expiration date that well. And then proof of structure doesn't mean you are going to get the same function. Usually for small molecule drugs, when you make a generic version of the small molecule drug, if you prove the structure, and then that be enough, and you can do only small bioequivalence studies to approve the biosimilar, the generic drug. However, in biosimilars, as I will talk about next, those are more complicated and also needs more control, and you also have to do some more clinical studies with them. So biosimilars, so it's you know, biosimilars are it's a big area right now. Companies are actually in a race of making biosimilar products, and the regulations of these biosimilars are just released recently by the FDA, US FDA, or the European agencies, EMA. So biosimilar is a biological product. Again, it's a biologics that's highly similar. So it's not identical. It's highly similar to and has no clinically meaningful difference from an existing FDA-approved reference product. Then when you say highly similar, it means it's a broad statement. So you have to prove it. Because FDA doesn't require your product to be identical, because it's almost impossible to have an identical biologic to a reference biologic. But you have to. The company job is to make sure that these small differences are not that critical for the product safety or efficacy. So the normal process for approval of a small molecule drug is that you go through discovery, preclinical animal studies, then you go through the clinical trials, phase 1, 2, 3, and then you get your drug approved. And then it goes to the market, and when it reaches the patent expiration, then then other companies will have the right to produce the same version of the drug through an abbreviated new drug application. So here, with the application of the generic drugs, you don't need to go through clinical trials. And then same thing with biomolecules. The biomolecules also go through the same route through the same pathway. And then, however, once the biologic drug reaches its patent expiration date, then recently, for example, the FDA, the Congress, they legislated a law that's called Biologic Price Competition and Innovation Act by 2010, which allows the other companies to make similar versions of the existing or the brand biologics, and they call them biosimilars. So biosimilars are kind of equivalent to generic products, but they are a little more complicated and more regulated, and also they don't have market exclusivity, just like generic products. So every company has the right to make a generic or biosimilar product. So so as a pharmaceutical analytics. Analytics actually makes the foundation of biosimilars. So for the standalone drug or the new drug or new biologics, usually it goes through the analytical characterization, and it goes through non-clinical and clinical pharmacology, PK, pharmacokinetic, pharmacodynamic, and then finally it needs the clinical trials to establish safety and efficacy. And guess what? The most expensive part of drug development are the clinical trials. These are super expensive. That's why biosimilars are intended to has low number of clinical studies, so that they reduce the price and so that people can purchase these products, in a reasonable price. However, for the biosimilars, the abbreviated version of the biologics, here the analytics or the analysis of these products, it makes the foundation of the approval process. Because if you can show as much as you can through analytics that your product is or similar to the brand one or the reference product, then the FDA people, they look at these evidences, and then they decide according of what you provide them. If your instruments are state of the art, very precise data, then they might ask for only a few clinical trials to tackle some residual uncertainties. Again, if you don't have, by the way, so the biosimilars, the cost of developing biosimilars is huge. It constitutes about 1/3 or more the cost of developing a new drug from the beginning. So it's 1/3. It's not like generics. Like I told you, Harvoni is $1,000 for a tablet. Like in India, they sell it maybe for $5 or something like that. So it's because the cost of producing Harvoni is a small market drug. It's not that huge. So while here still the price is high, but some biologics like for Herceptin or Humira, these are well established in the market. They prove themselves. So people, they don't want to develop a new biologics. So they want to depend on the existing success of these brand products to sell their products. Anyway, so as a pharmaceutics, and it's actually a collaborative effort, so we have to develop a new analytical techniques. We have to be very careful in terms of data interpretation and data analysis, so the equipments and the instruments are developing every day to increase the sensitivity and the precision, so that you collect as much as evidences to make this totality of evidence to convince the FDA to approve your product. So differences among biologics. So in general, this is a slide from FDA actually. So protein products or monoclonal antibodies as protein drugs can differ in at least 3 ways. So when you have a product, an protein product, or you have to make sure that your primary amino acid sequence is the right sequence, because these are living organisms. You are expressing your protein in them, so you have some proteases that proteolyze your protein, and also you might have your gene wrong in the first place, so you might not get the right amino acid sequence. So you have to make sure your amino acid sequence or primary structure is the right one. And also you have what's called post-translational modifications. Like protein is not only amino acids, it has also it's subjected to post-translational modifications. For example, you have the disulfide bonds. And these disulfide bonds sometimes are subjected to scrambling. They don't form in the right way. So you have to make sure these are have the right orientation. And the most important thing is the glycosylation pattern of your glycoprotein. Glycosylation is it's a carbohydrate, it's a sugar chain, usually attached to monoclonal antibodies in the CH2 domain, as I'll show you later. And this glycosylation or glycan is highly heterogeneous. So it's it's a chain, but you don't get a full-length chain. You get it's a mixture of chains. And these glycosylations or protein or glycoforms, they have different activities, different stabilities, different pharmacokinetics. So the goal is to monitor these glycoforms at least, or to hopefully make a uniform, homogeneous glycoform. And also the higher order structure, the secondary, tertiary structure, and also you have to test that, and because it's very important for protein folding and also protein-protein interactions. So let's take a look at the structure of the amino acid of the antibody in general, the IgG antibody. The IgG, it's in general, it's a Y-shaped molecule, and it's a homodimer. It's This is the monomer and a second monomer, and they are connected through the hinge region disulfide bonds, and also some hydrophobic interactions in the CH3 domain of the Fc region. So two Fabs responsible for binding to the antibody, to the antigen, and one Fc region which is responsible for what's called effector functions, for the pharmacological function, and also responsible for the antibody half-life. Antibody, by the way, antibody half-lives usually are unique. As I'll show you later, the biological half-life can extend to 21 days, which is very nice for treating of chronic diseases. It's a unique pharmacokinetic. And the most important part, I mean, at least from our perspective, because we studied this, is the glycan, or the sugar part, at asparagine amino acid position 297. They call it N-glycan, or the consensus N-glycan in the CH3 domain. Sometimes you have glycans here, but these parts are, you know, variables. It depends on the antibody. But this one is consistent within all the IgG monoclonal antibodies, and it's heterogeneous. So as I said, unique pharmacokinetics for antibody, for IgGs, or monoclonal antibodies that are based on IgG structure. So as I said, these antibodies have very long biological half-life, 21 days approximately. And the reason why, because they have a unique recycling mechanism. So the antibodies usually evade the degradation in the lysosomes through binding to a receptor called FcRn receptor. So when the antibody gets pinocytosed by the cell, or the degradation cell, it'll bind to this FcRn, and then instead of going to the lysosome and get degraded, it will go back or recycled to the surface of the cell, and then released and get saved from degradation. So how does what are modes of actions of antibodies? So there are several modes of actions for monoclonal antibodies. So the antibody, after binding the antigen or the target cell, can also bind through their Fc regions to an effector cell, which is usually natural killer cells, or macrophage or macrophages. And then by this binding, the effector cell will release some cytokines to kill the target cell through the ADCC activity. Another activity is through the activation of the complement system, and also to release some cytokines and form the membrane attacking protein to kill the cancer cells. Also, antibodies can get the effector cells, the macrophages, engaged to engulf and the tumor cells and kill it in this way, or antibody can have only a blocking activity, which tend to block the receptor ligand interaction and thus prevent the subsequent biological action. And again, another mode of action is through the ADC, antibody drug conjugates. The antibody binds to the certain antigen on the surface of the cell, and then the toxin will do the job by killing these cells. So these three mechanisms are collectively called antibody effector functions. So so what we are interested in is the N-glycan part of the antibody that I told you, I showed you on this CH2 domain of the antibody in the previous slide. So as we said, N-glycosylation is a fundamental post-translational modification, and it's heterogeneous. Why is it heterogeneous? Because it's not a template-driven process. So when you have a protein get expressed, you have a codon, and you have your tRNA, transfer RNA, and it has codons, and then the ribosome, and then this codons get translated into a certain amino acid sequence. And ما يقبل غلط, يعني it have to give you the right sequence. While in terms of glycosylation, this sugar chain, this process, it's not a templated. It's ماكو فد قالب يعني مقولبة highly processed. Therefore, the process can end here You have what's called MAN5, only 5 mannoses glycophore. It might end here, like it's to make what's called hybrid glycophore, or it might go all the way to the complex. And the complex also has different types. You have sialylated sialic acids, sugars at the end, or you have galactoses, or you have only glycanax. So when you have a vial of, let's say, Herceptin, if you really analyze this Herceptin, you don't find only one glycoform. You don't find only one structure. You get a collection of structures. Why? Because it's a mixture of different glycoforms. And these different glycoforms were shown to have different biological activities. So the problem with these different glycoforms, first you have, if you want to approve your drug, you have to make sure you have a consistent glycoprofile throughout all of your batches. And also, if you want to develop a biosimilar, it would be also a headache, because getting a certain glycoprofile, it means you have to use the same growth conditions, or almost the same growth conditions for your cells. And these these information are proprietary. They are top secrets for the innovator companies, so you cannot get them. So therefore, you have to do your best. Or you can stick, or if you can make one glycoform, and then you are done. But it's something impossible. No companies can reach that. So our goal was actually to make at least a couple of these glycoforms, a few of them, in a homogeneous way, and then study them separately. So that was the goal. And I don't think I can go through all the data today. But so in order to do that, you have to develop a synthetic platform that gives you homogeneous glycoforms. So what we used, So what we used, we took only the Fc part of the aminos of the monoclonal antibody, and here we use IgG2 subclass because it has only blocking activity. We need sometimes you need your antibody to have only blocking activity. Only you want to block a certain receptor. You don't want to recruit the immune effector cells. You don't want to have effector functions, because these are also sometimes it gives you some immunogenicity. So we made this IgG2 Fc in yeast. Since yeast, الخميرة, it has a Golgi apparatus, but doesn't. It has an endoplasmic reticulum, but doesn't have a Golgi apparatus. So what yeast can give you here, it can give you only high mannose or MAN9 glycoform, because it doesn't have the Golgi, right? So it doesn't give you the other glycoforms. So the idea was to make the MAN5 or MAN9 glycoform and then modify it in vitro using enzymes to make other glycoforms and study them in a systematic way. Right. So yeah, and then of course we need different enzymes, glycosyltransferases and glycosidases, to make these glycoforms. So we made these glycoforms, especially the fucosylated. Here is a sugar called the fucose. It's at the very beginning of the chain, and this fucose, has been shown to play an important part in the antibody-dependent cellular cytotoxicity. So recently, it has been shown, like, removal of fucose, taking off of fucose out of the glycan, it enhances the potency of the product by 50 times, 50 folds. And then, according to that, people developed a way to get rid of this sugar fucose through either knocking out the gene of the enzyme that adds the sugar, which is called the FUT8, fucosyltransferase, or using some inhibitors for that gene, for that enzyme, inhibit that enzyme to prevent the addition of fucose. Because if you have a fucose here, it prevents the interaction with the receptors or Fc gamma receptors on the surface of the T cells. While if you remove the fucose, then you get stronger interaction, and you go to higher biological activity in return. So our idea, we wanted to do the opposite. We wanted to fully fucosylate the antibody. We wanted to put as much as a fucose we can on that Fc of the region of the antibody. And in order to that, we had to make this enzyme, and also we had to make these substrates to make this. So basically what we do, we order the gene of interest, usually either the antibody gene or some enzymes genes. We made many of these enzymes, and then we clone it. We insert that gene into a suitable vector, and that vector then to make a certain plasmid, and then that plasmid get transfected to the inside of your expression system, which is either E. coli or yeast. In our case, use yeast because it gives you a glycoprotein, and then we express the protein, try to optimize the conditions to get as much protein as you can, because the most challenging part of studying these biologics is the proteins, making the protein, because companies usually they don't give you protein, and only R&Ds of companies are doing these types of research. So in order to compete with them, so you have to make your own protein. So and then after protein get expressed and secreted into the media, you have to harvest the protein, and using a couple of techniques like purification techniques, like some columns, protein G columns, HIC or hydrophobic interaction chromatography, and then you characterize the protein using different characterization techniques. And then what you do is you modify the glycan on that Fc, and then do the biological and the biophysical studies to confirm the protein physicochemical properties. And you do this redundantly, so that you eventually understand how this glycan affects the activity and stability of the antibody, and to eventually helps in the development of better antibody therapeutics. So as I said, we take the gene, put it in the vector, and then we express the protein. For example, then we run a gel. This is a purified protein, so you see two bands here actually. So this is the Fc. This is the Fc, and when you run a gel, SDS-based gel, the protein, you stain your gel, and then you get your protein, and you compare the protein to a marker, so to predict the size of your protein. So that was within the limits, or within the, however, it has some contaminations, it has N-glycosylated, doesn't have sugar, N-glycosylated Fc, so we had to get rid of this non-glycosylated Fc, and then you have to go through another set of purification techniques. And then after making the protein, then you have to formulate it in a suitable vector, suitable buffer, because you are not going to do your experiments in the same day. You have to freeze at least your protein. Usually we freeze our protein at either minus 80 degrees C or minus 20, but usually we prefer minus 80 because it's deeper temperature, and it prevents any dynamic movements of the protein parts, and thus prevents any chemical reactions and instability problems. For example, here, so if you look at this is a size exclusion chromatography. It's a column, it's just like a C18 or column. Then you run your protein through that to look for the aggregates, for the soluble aggregates. So the way that column works is that when you have aggregates, these aggregates will take shorter time and will get eluted earlier than the monomer. While the monomer, a non-aggregated protein will take longer time because it'll partition through different pores, and it'll take more time to get eluted out of the column. And then so this is one of the best techniques to monitor the protein aggregates, especially the soluble aggregates. So according to that, we screen through different buffers, and we do different couple cycles of freezing, thawing cycles, and see which buffer gives you the best and the lowest aggregation possible, so that you stick to that buffer and freeze your protein. And if you want to use your protein and you want to use different buffer to do your studies, then you have to dialyze your protein in the desired buffer so that you get rid of the storage buffer. And then I showed you the protein on SDS-based gel, but this gives you the approximate size. To get the accurate size or the mass of the protein, you have to do mass spectrometer. So as you can see, we made all of these, and we convert them by mass spectrometer. And it's actually amazing. So if you calculate the expected molecular weight and compare it with the observed, it's very close. It's like it's very close. So you go through all of these steps, and then you get the same molecular weight. So it's amazing. So you made this, and as you see, these are very homogeneous. It's only single peak, especially for the MAN5, and it's called fucosylated, afucosylated hybrid glycan, and then a fucosylated one, you see only a single peak. So that's great, because now you can compare this with this. This guy has only one additional fucose, which is a very small sugar molecule compared to the size of the antibody. And then at the same time, we made other glycoforms by truncating the sugar so that we get non-glycosylated protein, just the amino acid sequence, no post-translational modifications, and you use it as a control and see the. So this is our mass spec. As I told you, state of the art is the LC-QTOF. This is the LC system. It separates the protein, and then the protein then goes to the nebulizer to ionize the protein, and then the protein or the mist will go through a Q or quadrupole analyzer, and then it will fly through the TOF, time of flying tube. It will go like this and get reflected, and then go back down to the detector. And it's called QTOF, time of flight, because larger ions will take longer time to reach the detector out there flying on that tube, and that's how the mass spec detects the masses of different ions or different proteins. So, and then again, so we generated these glycoforms, homogeneous one. So the idea was to characterize them and do some comparative studies. So we did some turbidity measurements to see the stability of these different glycoforms. We did also far UV CD spectrum to probe the protein secondary structure, and some other techniques that I'll show you in a minute. So this is a turbidity measurement, just to measure the protein aggregation propensity. So as you can see, it's a pretty nice correlation between the size of the glycan and the aggregation propensity. So for example, if you have a high mannose glycoform, then this one will be resistant to unfolding at lower temperatures, and then it unfolds only at higher temperatures compared to the PNGSF digested or the aglycosylated FC that has no glycan at all. So it means that this asparagine 297 glycan is very important to lower the aggregation propensity of your protein and stabilizes it, because some other companies want to make aglycosylated monoclonal antibodies, no glycans at all, to delete any possible effector function, and that's not the case, because you would have stability problems. And also, then if you shoot your protein through the CD or circular dichroism to measure the secondary structure, you'll find that it'll give you your protein or the FC will give you a minima at 218 nanometer, which is an indication of a better rich secondary structure, which is characteristic for monoclonal antibodies. However, we do what's called CD melt. It's proprietary to our laboratories. So what you can do, you can monitor the secondary structure with temperature, and then you can calculate a TM onset, melting temperature, and also T onset starting point of unfolding of the secondary structure, and compare it among different glycoforms. And also another technique, it's called DSF, differential scanning fluorimetry. You mix your protein with a certain dye, hydrophobic dye, and then subject it to high temperature. And then once the protein gets unfold, the dye, it'll bind to these hydrophobic patches, and then it becomes fluorescent. So in normal cases, it interacts with water, it has no fluorescence, it gets quenched. But when the protein unfolds, exposed to the hydrophobic patches, the dye will bind to the hydrophobic patches, and no water of association, and it gets fluorescent. So again, you can measure melting temperature from this technique, and usually it gives you some indications of the protein tertiary structure. And of course, the DSC is the golden standard in studying the stability of protein drugs. So usually with the DSC, you got melting temperature again, and you heat your protein, and then you monitor the CP, or heat capacity, and you get a peak when the protein starts to unfold. It gives you what's called endotherm, and you see here two endotherms. The first endotherm is for the FC has two domains, CH2 domain and CH3 domain. So the first melting temperature is for the CH2 domain, and then so CH2 domain starts to unfold first, and while the CH3 domain will unfold later on. And that means that the CH2 domain has lower melting temperature, so it's more problematic. It's the CH2 domain where the glycan is. So if you take the glycan out of the CH2 domain, you see a vast reduction. It's like a 4.5 degree C reduction in the thermal stability of your FC region of your monoclonal antibody. However, the CH3 domain melting temperature does not change, because the glycan is far away from the CH3 domain. And again, this shows the importance of this glycan for the thermal stability of your proteins. And also, we wanted to study the effect of the glycan. The importance of this glycan is the sugar. So you would predict that usually sugars increases the solubility, because sugars are water soluble, so they would increase the solubility of your therapeutic or your monoclonal antibody. So it's hard to measure solubility in biologics. It's easy to do it in small molecule drugs, but even in small molecule drugs, it's very challenging. You have to reach the thermodynamic solubility, which is usually tricky. But here, in case of proteins, if you want to measure the solubility of protein, you need a lot of protein sometimes. So there are ways to measure protein solubility, like for example, take a certain protein concentration and then concentrate it using some concentrating tubes. But by the end, you need a very large volume of the protein until you reach a certain volume where the protein starts to aggregate. And then at that time, you filter your protein and measure the concentration and say, this is the solubility. But usually in drug development, product development, you screen, say, hundreds or thousands of hits, or they call them leads. So you cannot make a lot of protein at that stage. You can make only a small amount of protein just for to check for its binding to certain antigen, and then so at that stage you need really a high throughput technique to screen for protein stability. So we've developed a high throughput technique to measure the protein stability, solubility, and by using an assay called PEG precipitation assay. So in that assay, we use different concentrations of PEG, polyethylene glycol 10,000, and we put it in 96 well plate. And then we add a certain concentration, a fixed concentration of the antibody to these different concentrations of the monoclonal of the PEG solutions, and then we incubate overnight. And then guess what? If protein starts to precipitate out of the solution when the PEG concentration is high. So and then after that we spin down or centrifuge the plate and we measure the remaining protein concentration. And so in this case, so to make it easier, we usually plot the PEG concentration against the remaining protein concentration. So we get this kind of curves where the protein here solubility is okay, then start to decrease because the what PEG would do is it would withdraw the water from the protein, and the protein will get deprived of its water of association, and then it starts to aggregate. And then by using certain equation, the equation that I showed you previously, you can calculate what's called PEG midpoint, and you can also extrapolate to 0 PEG concentration. Like you go up, 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 and until you get the intercepts at 0 PEG concentration. This is what we call the apparent solubility, right? So the apparent solubility here, for example, and you get lower apparent solubility and lower PEG. And then we do the biologics, the binding studies, actually, where we use the Fc gamma receptors that I showed you previously, and these usually mediate the antibody effector functions. And again, we make these receptors in our lab. So this is a SDS page shows the protein after expression and purification to get it in a very pure form. And we use what's called BLI, bilayer interferometer, to measure the binding, the kinetics of the binding of the association of these proteins. So what we do actually is we immobilize our one binding partner on the surface of the sensors, and then we add the other the receptor to the solution, and then you get association curve, and then you get or KD rate, association rate constant, and you can also get the dissociation rate constant, and by this you can calculate the affinity. So we did that with these proteins, and we found that the addition of the fucose actually delete any residual binding to Fc gamma R3A receptor. And Fc gamma R3A receptor is an important receptor on the surface of natural killer cells. So it means if you add fucose, then you won't recruit NK cells, and which is good in our goal, because we don't want to activate these NK cells, because their activation is associated with some immunogenicity and anaphylactic shocks. And then this futate is the enzyme. We did some kinetics with the enzymes. We measured the KMs and the Vmax, and then when we found that the KM is different for free glycan versus attached glycan, while it's the same for Vmax, and then we proposed a model for the dynamics of the glycan. And then we also use proteomics to understand the glycosylation heterogeneity, because some people also they have a second glycan on the CH3 domain of their FCs, and beside the consensus or the well conserved glycan on the CH2 domain. So some people have another glycan on this, a few number of people. And then the glycoprofile of this glycan is different from this one, and then we had to understand why that was the case, and then we concluded that this glycan is buried inside the protein, so it's not accessible to some enzymes, like for example a futate enzyme. But nobody knew that before, whether it was an accessibility problem or some other problem. And then we did some kinetic studies, and we concluded that this glycan, we made a mutant, we made an FC which has only the second glycan, not the first glycan, and we did kinetic studies, and we found that the second glycan doesn't take fucose at all. That's why it's it doesn't have fucose from samples from human beings, or So it's just to summarize so what we did actually was successful generation of homogeneous IgG2-Fc glycoform using the synthetic platform that we developed, and also we found that core fucosylation, full core fucosylation, did not perturb the structure or change the stability of the FC, which is good from a formulation development standpoint. Also, we found that this glycan is indispensable. It's very important for the stability of the antibody and also the solubility. And also, we found that core fucosylation of IgG2 subclass reduced the binding to R3A receptor, which means it reduces the effector functions. And and sometimes you don't need these effector functions because they are associated with some side effects. And then from the kinetics, we find that there is a potential evidence that these glycans are more dynamic than the protein side chain, which could explain some of the points why the glycan is heterogeneous. And also some other kinetic studies, we understood why fucose is absent from the second glycosylation site. And so that was some of the data that I showed you previously. And also we might need another lecture for another day if Dr. Muwaffaq is interested or you guys are interested. And with that, I'd like to acknowledge some people.